Hello, everyone, and welcome to the Genomic Symposium. Today is a COVID-19 day, and in today's session, we will be discussing mostly our findings and approaches to understand the COVID-19 host genome. My name is Ravindra Kolhe. I'm a pathologist at Medical College of Georgia and Augusta University. And I'm going to introduce our group, our consortium, and introduce the speakers. As you know, COVID-19 pandemic has affected pretty much every part of the world. As I tell my kids, this probably is the biggest challenge we have faced since I was born, and we're doing pretty much each and every specialty in medicine is working towards understanding the disease and working towards getting the better outcome. As of now, more than 88 million people have been affected, and more than 1.9 million people have died. But one of the things which is fascinating about this disease is the split mortality. This is what I like to call you have this huge amount of individuals who are infected, but an extremely small percentage of them become symptomatic. And then in that symptomatic, really a small percentage end up dying. People have associated a lot of clinical comorbidities with the mortality. But again, even if you look at race, gender, age, or comorbidities, it's becoming challenging to explain all of these mortalities. We would have a younger population, less than 30 years old, without any comorbidities, dying, and in comparison to with the older population with the comorbidities. So as of now, majority of the meta-analysis have shown that the individuals who are hospitalized have 31% of diabetes, 50% of hypertension, and around 14% of heart failure. We know that the Black Americans are dying at twice the rate of the white Americans, and definitely the children, they tend to have a very low negative infection rate, but they do have their own category of inflammatory syndrome, which we call as multisystem inflammatory syndrome in children. And there has been a lot of global efforts in understanding the host genomics. Why do some individuals are affected worsely in comparison to the majority? As I said, there are multiple global efforts, and one of the biggest one is the COVID-19 host genome and genetics initiative. And we are being following the data from there. And one of the challenges which we identified based on the information which is already there public out of this initiative is these genetic studies are based on GWAS or whole exome sequencing, and they cannot detect larger variants. And then on the other hand, some of these variants, when we looked at it, which were identified, they were mostly severely associated with immune deficiency or immune defect. So we acknowledge what was happening around us, and one of the idea behind what we're doing right now was to see whether we can add to the existing knowledge base in understanding the split survival for COVID-19 individuals. So we get together, a couple of friends together, we're working, and we identified that there is a definite need and a knowledge gap with understanding the structural variants in these individuals who are severely affected by COVID-19. And we ended up putting together a group of labs who had a very similar interest, and we created a consortium called COVID-19 Host Genome Structural Variant Consortium. The idea behind this consortium was to put together a group of people who are working in the structural variant lab, so understanding the structural variants both in vertebrates as well as humans, and then see what we can identify in these groups. So some of the objectives of our consortium are, number one, we want to create a global networks of labs who use novel long-read molecule technologies for assessing structural variant in the host genome, with the idea that it could contribute to COVID-19 susceptibility or progression, because we do know some individuals post-infection get symptomatic and definitely progress to ICU or death. The second objective was to create a global networks of lab geared towards data generation and data sharing, mostly focused on host genomes, structural variant genomic data. The third objective of our consortium was to form a global forum where we can share this data which was generated among us from the different genomic methods, different bioinformatic tools, and different methods of interpretation of host genomic data. So it's not just one lab or one method of sequencing or one platform of sequencing. The labs which are part of our consortium come from different backgrounds with the different bioinformatic tools together. Lastly, the idea behind this was to develop a global guidance on data and knowledge sharing, again, mostly focused on host genome structural variant. We started with one lab at a time, and at this moment we have 35 plus labs across the globe, which are actively contributing to our understanding of structural variants in host genome for individuals who are affected by COVID-19. Each and every lab brings in a unique understanding of their understanding and knowledge of COVID-19 to the consortium, and it has been extremely fruitful collaboration with all of these individuals come together. We meet once a weekly over a call, 11 a.m. on a Wednesday Eastern time. I think we're changing that time based on availability. And then that has resulted in a pretty significant amount of data generation and knowledge base on the COVID-19 host genome. And that's what we plan to discuss today. Our approaches to understanding the COVID-19 host genome, as well as our initial data. So I just want to give you an introduction to today's speaker lineup. We have Dr. Sivash from UC San Diego, which is going to talk about the bioinformatics tools, independent bioinformatics tools for identifying as well as analysis of structural variants in the whole genome. Dr. Silvey from West Virginia, Commonwealth University, his idea about using bigger data in data mining bigger data, genomic data for identifying structural variants. Dr. Alex from Hoshin from Redbound, he had a major publication on structural variants in Europe. Dr. Michael Zodi from New York Genome Center, Dr. Eric Jarvis from Rockefeller University, Dr. Alan Becks and Dr. Catherine Brownston from Boston Children's Hospital will talk about the MIS-C syndrome in children. And at the end, I'll wrap up with our data on COVID-19 host genome, especially the individuals who were sick, symptomatic sick, admitted to ICU and died. So stay tuned for the speakers to get started. Thank you.